Good day, and welcome to the first of the 2018 AVC Winter Webinars, brought to you by the Sir James Dunn Animal Welfare Centre at the Atlantic Veterinary College in Prince Edward Island, Canada. I'm Dr. Alice Crook, coordinator of the centre, and I want to extend a warm welcome to all of you viewing this from around the world. This is actually a rebroadcast of Tuesday's webinar, February 27th, because I messed up during the recording of the webinar, and Dr. Overall very generously offered to redo this broadcast. Now it is my great pleasure to introduce her to you. So Karen Overall has Master of Arts, a veterinary degree, a PhD, and is a diplomat of the American College of Veterinary Behavior. She has given hundreds of national and international presentations and short courses, and is the author of over 100 scholarly publications, dozens of textbook chapters, and the texts Clinical Behavioral Medicine for Small Animals and Manual of Clinical Behavioral Medicine for Dogs and Cats, and of the DVD Humane Behavioral Care for Dogs, Problem Prevention and Treatment as well. She is the editor-in-chief for Journal of Veterinary Behavior, Clinical Applications and Research. Dr. Overall is a senior research scientist in the biology department at the University of Pennsylvania, where she studies the effects of anxiety and reactivity on performance and mental health in dogs. And she joins us today from Pennsylvania. So now I'm going to switch it over to your screen, Karen.
Excellent.
And there we go.
Can you now see that?
Yes. Yep. There's that kitten playing.
Yeah. So should I start?
Go ahead, please do.
Excellent. I love this picture because it's one of the things we're going to be talking about today. This is a cat doing very, very classic object play, and you can see this is a kitten. It's a slightly older kitten. And one of my big avocations and hobbies is great artworks, and I'll give lectures in places that have art museums that I hadn't seen before, because I'm fascinated by how people portray especially the animal world. And one of the things that I think is very important, because it's going to come up in this talk, is that if you notice this little kitten is using not just her feet, but she's using her claws to play with the dominoes. So that speaks to how cats perhaps should not be declawed, and what this means to the cat. But today what we're going to talk about is behavioral and neurodevelopment in kittens, and what you need to know to give them the best start and to minimize risk. And this is a long, detailed presentation with many neurodevelopmental charts, and we're not going to go into all of them in depth. Everybody should have access to a PDF of this. In fact, a PDF that doesn't have a number of the typos fixed that are fixed in this final version. But you'll be able to use it as a resource. And then if anybody needs references from me, I would be happy to provide them or to answer any questions. My email addresses are here. There are email addresses at the end. I do answer my emails, just not when everybody wishes that I did so. So I will be going through parts of this relatively quickly, and then I'll take a little bit more time to emphasize some of the things that I think don't usually get the attention they deserve. So these are the subjects about which we're going to speak. Evolutionary history of cats, which is very important, because if you really understand the evolutionary history of cats, pretty much every feline welfare and behavioral problem just sort of pops out at you. Sensitive periods in neurobehavioral development, and we'll be following a very similar pattern for dogs in the next lecture. Epigenetic effects on early development. Roles for temperament. People talk a lot about temperament, which is generally considered some stable behavioral attribute that is maintained much of the time through your life. It is not really quite as stable as everybody makes it out, but it's what makes certain individuals more outgoing and some individuals less outgoing. We're going to talk about paternal effects and social structure. We'll define social maturity and explain why it may be important for how we understand feline and canine and everybody else's behavior. We're going to talk about minimizing risk and meeting the needs of these cats and kittens. And then what I'd like to discuss is one example of a window into adult risk that will occur if we fail. And we'll just wrap up with some summary conclusions. So first, let's discuss the evolutionary history of cats. And I think the key thing to understand about cats is that early behavioral development in cats is affected by their evolutionary history. And cats are not small furry dogs. I know that people are now giving lip service to this, but they are not treating cats still as if they are truly cats. And I think that the key that most people don't even discuss is that cats are the quintessential sit and wait predator. This predation style has profound implications for how they play, how they react to scary and agonistic situations. You're going to see that it has profound implications for the types of food they eat and what that does to their metabolism. And you're also going to see that it has profound implications for how their brain is wired. And this then comes back and affects how they play and how they have pathologies. So cats were not truly domesticated as were dogs. Please remember that we domesticated stock about 12,000 years ago because they were useful to us. We wanted to largely eat them. And dogs may, in fact, not have actually been domesticated. They may have co-evolved with us. Dogs have been found in graves of humans that date to 30 to 33,000 years before present. And modern man has really only been around for about 60,000 years. So for half of that, dogs have been an integral part of our life. And if you look at the data that's out there and the evidence in places like the pyramids, there's evidence of different dog breeds and breeds that do specific tasks that's 10, 15,000 years old. So dogs have become more complex in terms of breeds, but certainly that split into different tasks was made quite early. We don't do that with cats. Okay, our relationship with cats is really based on a symbiosis or commensalism. And as humans became more agrarian, cats derived from multiple groups, like a wildcat subspecies, and they followed the migrating rodents out of Africa into the Middle East and across Asia as the agrarian societies began to move and take residence and stay in those places. So as we practice true agrarian agriculture, where we grew grains in places and we stored them, rodents, of course, followed the grains, cats followed the rodents. And if you look at the pattern of that, that all occurred about 9,500 years before the present. There were different founders and different variants of Felis sylvestris, but if you wish to look at the original paper, this is a Science 2017 paper, and it was by Carlos Driscoll and a number of feline geneticists, including Stephen O'Brien, who's done quite excellent and extensive work on the genetics of cats. And if you take a look at this, you can see that you have multiple founder populations, but you can see the spread and how it comes out of Africa, goes towards the Middle East. And in fact, pretty much all New World cats are derived, and near European cats are derived from the same group that came out of the Middle East about 9,500 years ago. So because of this commensalism or symbiosis, we actually did no selection for cats. They came along with us. Physical selection for coat characteristics is really only a few hundred years old. It's more ancient in many Asian cat breeds, but where we selected aggressively for work, and then coat characteristics came along with that in dogs, and then later we selected specifically for coats. We selected not at all for cats until pretty recently. And then when there was enough money for people to be able to have cat fancy and it became a status symbol, we began to select for what cats looked like. And in fact, you can track that in the history of artwork, because patrons who could afford to have paintings made of themselves often put their pets in them because you couldn't afford to have those pets unless you were wealthy. So you can actually do a lot of tracking of cat breeds and dog breeds through our work. Because the behaviors that we wanted in cats are those that were used to control rodents, don't forget, rodents would steal the grain, they would foul the grain, food soiling, and they carried disease. So anything that could get rid of the disease, the food fouling, and the food theft, because we're valued and because we wanted those behaviors, and they are the standard innate behaviors that cats exhibit anyway, we basically did no selection on human behavior. In fact, all selection on, I'm sorry, on feline behavior, all selection on feline behavior by humans is very recent. It's only in the past 50 or 60 years that you've begun to see people say, oh, we're advertising cats that are loads of personality, that are very friendly, that are very talkative. What they're describing are small dogs. They're not describing cats. But we've now looked for and selected many of those behavioral characteristics that we favored in dogs, and you now see people breeding cats that do more of these things. So we're all living through the period of feline domestication. Domestic cats likely have a ventral medial hypothalamus that reacts in a very characteristic and abrupt way that recruits neurons as a result of this innate foraging style. In other words, because they are sit and wait predators, and they have to go from sitting very quietly and being very attentive to pouncing almost instantly and then engaging in a struggle to kill a prey. And don't forget, they eat multiple times a day, small prey sources. They're solitary hunters. They don't work in groups like dogs do. But as a result of that, we have this very bizarre ventromedial hypothalamus and lateral hypothalamus that react in a very characteristic and explosive way. And in fact, these regions of the brain recruit neurons so quickly and in such a stereotypic manner that cats are often used as animal models for epilepsy, because that recruitment of neurons is commonly called kindling. And that's a great model for epileptic seizures. So that is a neurological adaptation to the sit and wait foraging mode that again carries with it being a solitary predator, not solitary socially, a solitary predator. And you have to be very still and then be able to become very fast very quickly. And then we see that the effects of that appear again later in pathologies. In fact, the blueprint of our lack of physical and behavioral selection in cats can be seen in two data sets. We see it from the neurodevelopment of cats, not just the ventromedial and lateral hypothalamic cell structures and reactivities that I just mentioned, but the neurodevelopment that we're going to discuss that are derived in part from this evolutionary history. But we also see it in some interesting physiological history that's associated with hypercarnivory. Excuse me, our main focus is neurodevelopment, where we see absolutely no selection for extended sensitive and social periods, and where we have quite short and easily and early truncated periods for social and physical exposure, which is very, very different than we see in dogs. And next week, when we talk about dogs, this will become clear. But one of the things that domestication has done is to damp down extremes and to spread out the plasticity of neurobehavioral development, and to spread out those time periods and tails so that the individuals are more able to interact with humans. Domestic dogs are very like humans, which have these long and extended social periods, but wolves are very much like cats. In other words, wolves have very narrow windows of exposure to other species. And if you wish to handle a wolf at any point in its life, you best handle it before 14 days of age. So we see that residual, inherent, ancestral, safe, truncated period still remaining in cats, and it is not remaining in dogs. And that's another piece of evidence that we haven't truly domesticated them. Dogs, like humans, do well as omnivores or mesocarnivores, and that pattern is likely affected by their relationship with humans. It made them easily attracted to humans because they could take advantage of human garbage. Wolves today still forage on human garbage. Foxes, all carnivores will forage on human garbage to some extent, but the canines, whether it's a coyote, a fox, or a wolf, will all do it. But what we tend to see is that if there is no immediate access to large prey that you can take down as a group, certainly dogs and wolves are capable of foraging on lots of things, including fruits and vegetables and leavings. And that's not a pattern that you tend to see in cats. And in fact, if you look at the effects physiologically of hypercarnivory, there's been a big phylogenetic shift. And this is a paper that's in PLOS One. It's an open access paper, so anyone can get it. And this group looked at mutations that occurred multiple times throughout the history of the development of felids. And they looked at carnivore phylogeny and UGT mutations and diet. And if you take a look over here at what I've circled, you see the domestic cat. And 6.2 million years ago, when it split from the leopard cat, there was a base pair deletion in this UGT 1A6 enzyme. And when you understand that the UGT system is the system that's responsible for the metabolism of phenolic compounds, you begin to understand that this is one of the reasons why cats don't metabolize drugs the same way dogs do. This is a great adaptation to hypercarnivory, where you never see plant substances. If you see plant substances, you need to be able to break down phenolic rings. But what you see is the cats do this step, the UGT step, very badly. And it's essential in the metabolism of many drugs. These are just benzodiazepines, and these are the primary metabolic routes. But there are secondary metabolic routes to diazepam, for example, that also can involve UGT enzymes. The mutation that was looked at, don't forget, was for the UGT 1A, and it was the 6. But that doesn't mean that we don't see the same patterns where you don't metabolize things as well, and you have very long half-lives in cats and all the UGT enzymes, because they all affect things like bilirubin metabolism and the metabolism and breakdown of phenolic compounds. So that ancestral feline blueprint shows up in how you metabolize drugs. So there are similar fingerprints for the characteristics that define feline behavior all over their neurodevelopmental and sensitive periods, which is really the main focus of this talk. So I need to introduce to you the concept of a sensitive period, and a sensitive period is best defined as a period when animals can best benefit from exposure to certain stimuli, and if deprived of such exposure, there's an increased risk, okay, this is a risk assessment, an increased risk of developing problems that are attendant with that stimulus. This does not mean that if you absolutely miss a certain period, you will absolutely positively have a problem. What it means is that that period, if you can have the exposure, is meant to give you greater plasticity and decrease your risk of developing a later problem. If you lose that plasticity, you enhance the risk that you will have later problems with a lack of that plasticity. Okay, so when animals are neurodevelopmentally able to respond to certain stimuli, they benefit from exposure. The problem is not every animal is ready to the same extent at the same time. So what we need to do is make sure that these stimuli are available in non-threatening and relatively benign contexts, so that when the animal is ready to be exposed, they can best benefit from that exposure. And lack of exposure will then increase the risk that they develop behavior problems associated with the omission. For example, if cats never see people before they're 2 months of age, they're always going to be reactive to people. And that doesn't bode well for them having a good and humane existence. Okay, let's talk about the neurodevelopmental landmarks of cats. There are half a dozen of these charts throughout the next 10 or so slides, and I'm not going to go into all of them in depth, but I will pick out certain hallmarks, and I've highlighted in pink the ones that can be associated with specific problems. I want to point out some seriously odd patterns for you that you'll see in cats. For example, at day 24 of gestation, if you look at the first comment up here, and let me get my pen going. If you look at tactile sensitivity, okay, day 24 of gestation. Vestibular writing reflex, day 54. Purring begins 2 days. The age at which separation from the mother leads to fearful and aggressive behavior to both cats and humans, 2 weeks. So if they're separated from mom at 2 weeks of age and don't have that tactile exposure and that nourishment, you're already looking at a cat who's more likely to be aggressive to humans and cats. We know there are temperature influences on how well they thermoregulate. And if we look at the next slide, we know that how close another kitten is physically between 2 to 4 weeks indicates how calm the cat is. So this is always a concern for singleton kittens or for orphan kittens. And the solution is to make sure they have access to other cats and to kittens. But if you have this physical closeness, if you have this tactile stimulation that's pressing, you're likely to be calmer. Singleton kittens emerge from a nest box a little earlier than non-singleton kittens. And if kittens are exposed to another species by 4 weeks of age, 4 weeks, these cats are not out on their own yet. They will show no fear of that other species at 12 weeks. Let's go back and look at some of these other dates because they're important. At 3 weeks of age, mom will begin to teach predatory behavior. This is interesting because she's still stimulating the kittens to urinate and defecate at this point in time. So they still need her to help them with elimination functions, but she's beginning to teach them to hunt. That tells you a couple of things. It tells you that these periods are short and they're truncated and they're jam-packed as a lack of domestication. And it also emphasizes how important the ability for these cats to become early solitary predators is going to be for that sit and wait mode of hunting that is common in these small predators. Okay, cats cannot retract their claws until they're 4 weeks of age. So obviously teaching them to play nicely with their claws until then is a good thing to do. Please remember that moms move kittens through the first month or so of life, and that during that period, kittens are beginning to learn to play. And we're going to talk about that in a lot of depth. Kittens will choose a place to eliminate sometime between 5 and 6 weeks of age. They may not cover their urine or feces, they won't dig, but they will choose a place to eliminate. Before that, they'll just lie in that substrate starting at 3 weeks of age. And it's no small shock to people if they think about these cats coming out of Northern Africa and going throughout the Middle East to realize that cats innately choose open, reflective, well-drained substrates. Well, that's sand. So, light-colored things that are reflective, that are well-drained, that let urine flow through, and that allow feces to dry. If you handle kittens between birth and 45 days of age, and we'll talk about what we mean by handle when we talk about these data in depth, they will regularly approach unfamiliar objects rapidly. So in other words, human handling transfers to object handling of inanimate objects. And they'll certainly spend more time with them at 4 to 7 months of age when they get older. So that's important because inanimate things are not the same as animate things. So by interacting with the cat at a very young age, you are enhancing more than just the social regions of the brain. If kittens are handled by numerous people between 5.5 and 9.5 weeks of age, they show less fear and are more interested in people later. And interestingly enough, the age at which they begin to respond to olfactory challenges is also the time that they begin to cover their urine and feces if they are going to. Now, they needn't cover their urine and feces. And in fact, the ancestral condition is to leave it open because you're signaling a lot about hormonal states and what you ate and where you live and how frequently you come back and health status. And because of parasite control, you would very much like for those feces to dry out. So if cats don't cover their urine and feces, that may be normal. If cats that did cover their urine and feces stop covering as an adult, you may wish to ask the people how often they're cleaning the litter box and if they're meeting the cat's needs, because a change is different than a pattern that was a normal pattern that just wasn't what the clients were expecting. If you then look at how social play develops, it continues through 7 to 10 weeks of age, and you see there are some differences in how males and females play and how big they are. During the first 2 months of life, the social relationships that you make, if you're going to live in a social group of cats, are those that will determine who your offspring will nurse. So cats do communal suckling. Some of it's nutritive, some of it's not, some of it's neurodevelopmental and companionable and is about emotional status, but you learn the individuals who will communally suckle your offspring and whose offspring you will also suckle in the first 2 months of your life. By 2 months of age, object play has dramatically increased, as in that picture we showed at the beginning. And you see that different types of play behaviors, like standing up and pouncing, are 90% effective at getting a play response by 6 to 12 weeks of age. But by 12 weeks of age, you're shifting from social play into more social fighting. Social fighting really starts at 14 weeks of age. You see real sex differences in social play as you're moving into the social fighting period. And by 12 to 14 weeks of age, social play is declining greatly. Feral cats completely abandon kittens as early as 4 months of age. And that's interesting because if there are adequate resources, and we'll discuss this later, cats will stay in their native cat groups for 18 to 24 months of age, even if they're males. They may not reproduce on their own. They may help her other kittens. Certainly, if they're females, they'll stay there their whole lives. So it all depends on the resource environment and the relatedness of the group. Okay, the earliest under well-fed, well-managed, non-stressed conditions that social weaning from the maternal group usually occurs is 10 to 14 months of age. So if you're a feral cat and you have not had exposure to humans, and we'll talk in a minute about the effects of the nutritive environment, you may, as early as 4 weeks of age, be thrown out of that group. That's a big, 4 months of age. That's a big difference. You're talking about a 6 to 10 month difference. So this matters when we go to talk about the welfare of feral cats versus the welfare of pet cats, because they're not the same cat. And they have different concerns, and we need to understand this neurodevelopmental pattern if we're to prevent some of the worst effects of being a feral cat, and if we are to, in fact, to handle feral cats humanely. So this is just to remind you that females teach their kittens to start being predatory at 3 weeks of age, and by 5 weeks, the kittens can kill mice in their own. I know this is an adult cat, but I wanted to drive home the point that one mouse is a meal for a cat, and you see why that sudden grab, pounce, bite, deliver the killing bite becomes so important. Okay. Excuse me. Males engage in twice the object play that females do by 7 weeks of age. By 7 to 8 weeks of age, you see eye-paw coordination really honed quite well. This is a slightly older 7 to 8 week old kitten, and you can see it really, it's tracking up this toy. It's grabbed the end, and now you can see this little kitty from a shelter tracking all the way up the toy. These are younger kittens. These are kittens that are only 5 weeks of age, and they don't quite have the eye-paw thing down, and they don't quite have the social thing down. So 2 weeks of age here, and you can even see it in a photograph. By 60 days or 8.5 weeks of age, object play increases. By 6 weeks of age, sidestep posture elicits play 20% of the time. And by 12 weeks of age, it's down to 3% of the time. So here's this little sidestep posture here where they step off and the other cat will then interact with them. And it very much depends on what age they are, what the response is. This pounce, belly up, stand up display between 6 to 12 weeks of age is 90% effective in obtaining a play response from another kitten, which would doubtless happen here, except this little kitten is following a trail of treats. By 12 weeks of age, again, social play patterns become more associated with social fighting and predatory behavior, as you see here in these older kittens. They're fighting outside. And you see these behaviors pertain to true social fighting as cats get older. And you'll see some of the same behavioral patterns in cats who are having pathological aggression between cats in the household that you would see in older kittens. Okay, I've put this chart in there for those of you who are interested in play, and it just outlines all of the developmental play behaviors in cats. I find that this is an extremely helpful thing to give to and discuss with clients because they really don't understand cat play. They don't understand the evolutionary history of cats. And if they begin to understand these things, it's amazing how fascinated they become and how passionate they become about meeting the cat's needs and interacting with them. Okay, let's talk about epigenetic effects on early cat behavior. It's interesting because all of the data I'm going to show you on nutritive effects in early cat development were done in the 50s and 60s and as late as the early 70s. The concept of an epigenetic effect, however, is a new one, although it applies to those data. Epigenetic effects are what happen when you have glucocorticoid excess pre, peri, and postnatally with learning at the cellular and the molecular level. What ends up by happening is if you stress the individual either when they're pregnant or the baby pre, peri, or postnatally, either because of social stress, physical stress, or the mom's nutritional stress, you now get a spike in cortisol. Chronic glucocorticoid exposure actually affects the development of the hippocampus. This is the region of the brain involved in associative learning. So in other words, we tend to think of it in terms of verbal signals and a behavioral response. So the easiest social learning pattern is asking your dog to sit. Sit, good girl. There you go. So that's an associative learning pattern that occurs in the hippocampus. Chronic cortisol elevation appears to act as what's called a translational gene regulator and a hormone response element. And cortisol itself, if it's elevated, acts as a hormone response element, and it interferes with your ability to learn, even that is simple associational learning in the hippocampus. Because in order to learn something and to retain that learning, you have to make new protein. And hormone response elements like glucocorticoids actually block the translation of the encoding of that protein. Forget the epigenetic effects. That happens simply as a result of the cortisol, so that you cannot learn something new because you cannot make the protein that would go with the learning. We get to the epigenetic effects when we look at prenatal and chronic ongoing stress, and most of this work has been done in rats, but it's also been shown to be true in humans. It looks like it's probably true in dogs and cats. The research is still lacking there, but so's the funding. But I do think it would be worth doing, and I've been pursuing it because our data show that it's turning out to be far more important than we thought. With this ongoing stress, you get lower levels of extinction of cue conditioned fear. You get shrinkage of the hippocampus and subsequent memory impairment. You get facilitation of fear conditioning in the amygdala. The amygdala is the part of the brain responsible for learning fear. You need to learn fear as an adaptive response to stay safe. But if you have an overreactive amygdala, you now become fearful of everything. Remember what we said about cats who lose their moms or who are not exposed and how fearful and reactive they become to things. That's an amygdalic response. So that stress response facilitates fear conditioning, and it's especially potent for any fear conditioning involving sound or auditory fear conditioning. And of course, some genetic backgrounds protect you and some genetic backgrounds put you more at risk. So here's what I mean by you need to make protein to learn a new behavior. So here's a human brain. This is the amygdala, this yellow knob. It's the end of the hippocampus, and these are paired structures. You can't see the amygdala on the other side, but it's about here. The red is the hippocampus, responsible for associative learning, and the amygdala is responsible for reactive fear, learned fear, adaptive fear, and when it's really overly stimulated, especially in this epigenetic state of high cortisol arousal, you can learn to become fearful of anything for which you make an association. And what ends up by happening in learning is you get all sorts of synaptic inputs. They could be drugs, toxins, learning. It could be anything, behavior mod. You stimulate neurotransmitters, you stimulate ion channels. Those stimulate protein kinases and transcription factors. You turn on a region of the gene. You then make new proteins in the coding region. Those new proteins, through messenger and translational RNA, then become new neurotransmitters, new ion channels, et cetera, and that contributes to your behavioral phenotype. So what ends up by happening when you're in this epigenetic stress environment because of glucocorticoids is you've decided you're going to learn something. So you're going along and you're translating the protein and you're translating the protein, and CREB is translating it. CREB is cytosolic response element binding protein, and you need it to learn things. And it goes along and it's like, I can make protein, I can make protein, I can make protein. Oops, there's your glucocorticoid receptor acting as a hormone response element, and it stops translating it. So not only are you more reactive because of the epigenetic effects, but now you can't learn anything to compensate for that. So let's go back to the old data now and see how this fits with new theories of epigenetic factors. And if you're interested in epigenetics, perhaps the person who's doing some of the best work in the world is Bruce McEwen's lab at Rockefeller University in New York. And if you look up any of their papers, they're utterly fantastic. The work is rodent work because it's the affordable work to do, and it's the model system. Okay, if you early wean cats, object and other types of play appear really early, way before the 2 months of age. Remember we said 60 days, way before that. If mom is fed 50% of ad libitum intake during the second half of gestation, so the second half of her pregnancy, first half could be fine, the second half isn't, and 6 weeks postpartum where she would get to where she's nutritionally weaning the cat, you get abnormal play, more aggression in males during social play, more climbing and running behaviors in females. You get slowed growth of the cerebrum, cerebellum, and brainstem. So you're actually, for the first time, seeing a neurodevelopmental effect of nutrition on regional brain growth. If queens are fed 50% of intake at gestation, in other words, throughout the whole period, Kittens have delays in postural corrections. They don't walk normally. They don't climb normally. They can't fix it. They have delayed predatory and exploratory behaviors. They're tiny and they don't learn well. They've got increased reactivity. They have abnormal fear and aggression and decreased responsiveness to the normal environment. We'll take a look at this. This just described feral cats. And how well do you think most feral cats, especially in bad urban environments, are fed? Chances are they are only getting about 50% of their nutritional intake. And it's not surprising that we have problems with these feral kittens in many environments. Low protein diets late in gestation and during lactation are responsible for delayed development. This is protein, not overall calories. This is intake period. Okay, you get uncoordinated and fewer social interactions with moms and poor attachment responses to mothers. That means that in the next generation, you're not going to do that communal and affiliative suckling and caretaking. That means that you're not going to learn to do those normal social behaviors that mom teaches. You're not going to learn normal predatory behavior. So now you can see how you've got this evolutionary history nested in these already truncated behavioral periods, nested in stress, nested in nutrition, and they all affect the outcome and the welfare and the well-being of these kittens. Okay, remember what we said is some genetic environments are protective and some put you more at risk. Well, this is where the concept of temperament and boldness and shyness comes in. So let's talk about boldness and shyness and exposure to humans, given these short and sensitive periods, and ask ourselves how these might differ from dogs. Well, next week, you're going to be able to answer that question for yourself. But let's look at the important periods of behavior in cats. And this is data from Karsh Turner. Dennis Turner is an ethologist. Eileen Karsh is a psychologist. And they did this work in the 1980s. And as we already know, mom teaches predatory behavior at about 3 weeks of age. The cats can do it themselves by 5 weeks. You saw that picture of a cat with a rodent in her mouth. They respond to social and olfactory threats by 6 to 8 weeks. They are nutritionally weaned. In other words, they're not really getting all their calories by suckling by 7 weeks of age, which is also when eye-paw coordination increases. And we know that by 14 or so weeks of age, social fighting is established. Until then, we can consider the social play period. And Karsh and Turner did a series of very clever experiments where they broke the social play period into 3 periods, the early, the middle, and the late period. And these are the periods to which we're going to refer in the next few slides. So the early period experimentally is what they called the 3 to 4 week period. The middle period, 4 to 7 weeks. And the late period was 6 to 10 or 12 weeks. So there was some flex here. So group 1 here is the early period, 3 to 4 weeks. Group 2 is the middle period, 4 to 7 weeks. And group 3 is the 6 to 10 to 12 week period. And yes, I know there are overlap in those groups, but this is the cats they had in front of them. You know, research is dictated largely by the practicalities of life. And their first experiment looked at the effects of handling on staying and approach latency for cats that were handled 15 minutes a day. So in other words, they had these kittens in a laboratory and they were bred, and they would pet the kittens very gently. They would take them out of their cages, gentle them in their hands, and just very slowly pet and massage them for 15 minutes a day, and then put a chalk mark on the kitten and put it back in the cage and move on to the next kitten. And they asked, if someone then picked up the cat, how long would that cat stay with the person, given the period of time in which they were handled? And how long would it take that cat to approach a person, given the period of time they were handled? So for the cats that were handled at 3 to 4 weeks of age, they would stay with people for about 41 seconds. And they were pretty quick to approach people. If you delayed the handling, this is where you delay the handling to 4 to 7 weeks of age. And what you see is now this is increased a bit. in the duration of stay, the latency is really increased. And if you delay the handling some more, it's an interesting pattern. You get a duration of stay that is less, but you also get an approach latency that's much greater. So waiting to handle cats to 6 to 10 to 12 weeks of age is really not a good idea if you want a very social cat. Well, that makes sense because they're now out of that sensitive period for playing and interacting. And these pairwise comparisons are all statistically significant. So let's go back and just pay attention to groups 1 and 2. Notice that the Group 1, 3 to 4 weeks of age, is the period where you stayed around longest and your approach latency was the shortest. If you then looked at 4 to 6 weeks of age, you decrease the amount of time you stayed and your approach latency, the amount of time it took you to approach somebody, increased. What happens if we just look at these two groups and we increase the amount of handling time to 40 minutes a day? Well, look what happened. Suddenly, these cats stay around really a long time, and it doesn't take them very long to approach somebody, and they are indistinguishable. So by increasing the time early, you can have both a multiplicative effect on both of these factors, and you can diminish the risk that the second group would have felt by a slightly later start. So early starts and enhanced handling time can really help kittens, especially if they're orphaned kittens, if they're wild kittens who might come in who may or may not be feral, if they're cats with an unknown history, kittens with an unknown history. Bear this in mind, 40 minutes a day. So just to remind you, between 5.5 and 9.5 weeks of age, if kittens are handled by numerous people, and there were only 5 kittens in this one study, they showed less fear and more interest in people later. And you see a kitten in this age group who just didn't have that advantage. This is a shelter cat, and he's terrified of absolutely everything. Okay, so now Karsh and Turner did a further study. And this is the study where boldness and shyness matters. If you look at handling at weeks 1 to 5, remember this is before that early period and the early to mid period, weeks 2 to 6, early to mid period, weeks 3 to 7, late early through late mid period, and then the later period. And you looked at all cats in their study, and you looked at, this is just how long you can hold on to a cat. If you reach for a cat and gently pick it up, how long will that cat tolerate being held before it pushes to get away? If you start very early and stop relatively early in the middle period, it's just like starting late, okay? 87 seconds. If you get them through the early to middle periods inclusive, you can hold them 30 seconds longer, and that's a statistically significant effect. If, however, you break your group into non-timid cats and timid cats, bold cats and shy cats, if you look at the bold cats, you get the same pattern. So early and late are not nearly as good as getting the entire early and middle periods. So you get the same pattern on either end, but you get a multiplicative bounce from these being bold cats. So bold cats get more out of this than cats on average do. But here's where this becomes really interesting. If you look at these timid or these shy cats, now granted, the sample sizes are small, but this is one of the best examples of a gene-environment interaction in the literature, and almost no one cites it. This is flat. This is absolutely flat statistically. There is no difference between any of these groups. So not only do you not get this pattern where early through middle periods really makes a quantitative difference, but you get a flat period throughout, indicating that the risk is far greater for an individual who would be shy anyway. Now I want you to ask yourself what the chance is that individuals who have been dumped or become feral are shy and don't get along with people, or are fairly reactive because of epigenetic effects and don't get along with people. And they are dumped back into that feral world, and then they reproduce and maintain the cycle. I want you to think about that, because they're not the same cats and they're not the same populations. Meanwhile, if you get a bold cat, and this is a slightly older kitten in one of the labs we teach at the North American Veterinary Conference Institute, which is a great course, and I'll tell you more about it later, but this is one of the interactive labs. And here you've got this bold kitten who's chasing this feather toy up a scratch post, and then he chases it over to the chair, and then he jumps back to the scratch post, and he did this for about an hour. Absolutely charming, interactive cat. That's the cat everybody wants. Okay, what about the effects of dad, and what about the effects of social structures? So if we look at the effects of dad, there are a series of papers that don't get a lot of attention, but are out there. And I'm showing you the original abstract because this is a 1986 paper. There's always something to be said for reading the literature. We don't do it enough anymore. But when the authors, who are Bateson and Turner and a few other people, when they looked at cats in lab situations where they knew the fathers, and these were not in a building lab situation, these were cats that they could contain more or less, the extent to which they were more or less friendly depended on who their father was. And they had two fathers, and they knew who was friendly and who wasn't, and the friendly fathers had friendly kittens, whether or not the fathers stayed around. And in fact, the fathers did leave, because the model for absent parenthood in fathers is cats. So these dads didn't have to be there to make their kittens friendly. Well, that sort of makes sense, because Sharon Crowell-Davis's lab has done a lot of work on the social structure of domestic cats, and they found out that they live in large extended matrilineal groups with an extended and extensive female group that provides care and shared care of offspring. Somewhere at about 10 to 14 months of age, if the resource environment is dicey, you get some bachelor males. The males are the ones that disperse. If the resource environment's good, those guys could stay around for a couple of years and help raise young. If mom was feral, or the kittens are feral or orphaned, obviously these become bachelor males quite early, and they are not quite normal. Sexual maturity is about 6 months of age. Social maturity is very, very, very delayed in cats if the resource environment is great. And it may be delayed anyway, because although they are solitary hunters, they do best if they live in colonial groups, because they provide care, they provide protection, they provide a lot of things that you otherwise couldn't have if you didn't live in a group. And we'll talk about what social maturity means, but generally there is a lot of compelling evidence that this doesn't occur until 24 to 48 months of age in cats. It's a little earlier in dogs, but there's a big role for the resource environment. We need to be mindful of the fact that cats are induced ovulators, and that if people have catteries or they have feral cat colonies they're helping to maintain, that if a new male takes over because they're induced ovulators, that male may kill any suckling kittens so that the queen comes into season and they then can impregnate her. The evidence for this comes from wild lion prides, but it has been shown to occur in some domestic cats. And again, the heritability of the boldness via the father, Sandra McCune has substantiated this. As I said, Turner and Bateson have substantiated it. A number of people have looked at it. Let's talk about the social maturity thing, because it's another thing that makes cats very, very odd. What's social maturity? Well, in humans, social maturity is a period of renewed but progressive myelination, and then some regressive pruning that is associated with changes in neurochemical profiles and in changes in developmental behavior. And we have no reason to believe this is any different in dogs and cats. As humans age from 6 to 17 years of age, the gray matter volume begins to decrease and the white matter increases, and the connections between the hemispheres of the brain get greater, and the corpus callosum increases in mass and in volume, suggesting that this integration is exceedingly important to aging. So that happens at 6 to 17 years of age. Then we get into the further myelination and then the regressive pruning, because in order to do that, you grow new neurons everywhere. Well, you don't need all those neurons. What you need is an efficient communication network. So in studies that have been done at the National Institute of Health in the United States, cohorts of humans were assayed through standard MRI, and males and females were followed in annual cohort groups through 27 years of age. And what was found was that connections between the frontal and the temporal cortex seemed to develop more slowly than other regions of the brain. So that's important to know. And it shouldn't surprise you to find out that when they stopped this study at 27 years of age, all of the females, in fact, had pruned their neurons. There was still a large proportion of the males who had not finished pruning their neurons, because as we now know, males have much more prolonged tails to their social development period. Patterns of change and maturation of the human frontal cortex, where you prune the neurons and you organize them more efficiently, improve cognitive processing. And we now know that that's not just supported by the MR work, but through electrophysiological PET and neuropsychological studies on cognitive and neurological development. We know that trauma affects this, and we know that early epigenetic affects whether or not you prune correctly. So this is another reason to make sure you have a good nutritive and social and developmental environment. And unfortunately, the imaging studies are completely lacking for dogs and cats, but they're possible. I've tried any number of times to fund these. They're extraordinarily expensive to fund. No one sees the need for it, but I think inductive reasoning tells us that this has to happen in dogs and cats. What we do know about cats is that they seem to enter social maturity later than dogs do, as a correlate of that matrilineal system with the shared care, and they seem to go through a real apprenticeship phase. Don't forget, they have to hunt in a solitary manner. Dogs can hunt in a group, so the apprenticeship is very different. Cats are likely socially mature by 2 years of age, but we see the patterns spreading out over the first 4 years. There is other evidence that supports this. Females reach their adult weight only at 2 years. Males reach it at 3. There it is again. The males are developing later in every species that's being looked at than do females. Lowe and Bradshaw reported that between 4 months and 2 years, temperament measures shifted more than is commonly accepted. In other words, temperament wasn't as stable as one would have predicted. That makes sense, because when you're pruning these brain neuron regions, things can go wrong, they can become destabilized, they can get worse before they get better, or they can go completely off the tracks. These pruning periods are a time of great risk, and they are when we see the incidence of psychiatric disease begin to blossom. Dogs appear to enter social maturity as early as 10 months of age, and they emerge as early as 18 months, but the ranges seem to be 12 to 36 months of age. People notice that between 12 and 18 months of age, dogs who were tested in a repeated measures way on cognitive tests or performance tasks seem to begin to have a more standardized performance than if you looked at it before 12 months of age. But it is definitely through 12 to 18 months of age, which is why we don't tend to put dogs into work until at least 18 months of age. The period of social maturity has never been well measured behaviorally, physiologically, or neurochemically, or through functional imaging in dogs and cats, but I have no doubt that if we do so, we will have similar findings. And I think that we have to realize that given the shared selection pressures on the development of behavior, we should expect dogs and humans to experience similar age-related developmental brain changes. Okay, now, remember, development. So at 8 to 10 months of age, male cats stop squatting and start spraying. They're not socially mature yet, but they're sure sexually mature. That's just like us. We're sexually mature before we're socially mature. And unless they're stopped by making marks by something in the social environment, they'll use their claws, they'll use their sheaths, they'll leave olfactory marks, they'll use urine. There are lots of ways to do this. Okay. Let's finish up here and talk about, now that you know all of this stuff, how can we minimize risk and meet needs? Can we meet the neurodevelopmental needs of cats? Can we do that? Can we, as veterinary professionals, make recommendations that will help people meet and enhance the neurodevelopmental needs of cats? Well, can we provide good dads, friendly fathers? Can we provide well-nourished, non-stressed moms? Can we look for bold parents? Can we have as early as 2 weeks, consistent and kind handling from humans? Can we have early exposure as early as 2 to 3 weeks? Remember, moms move kittens a lot to new and stimulating environments. Can we meet the food needs of cats? Remember, classic sit and wait predator, solitary hunting style, affects activity for food, brain reactivity, and pathological aggression when it happens. Remember that we have done nothing to change those hypothalamic reactivities. Size and social organization mean that group hunting doesn't occur. Instead, pathways that prey could pass are controlled and fast, and fierce arousal really matters to these cats. Both claws and teeth are used. Prey is the size of one meal and may be shared with dependents, which could be babies, the ill, very old, and nursing. Remember that communal care. This is very important for cats. And we really meet none of those needs with a bowl of cat food. There are loads of ways we can do it, though. These are some of the commercial toys on the market. This is the most expensive of the group, a Pipolino, where you can adjust the size. And this is lovely because then multiple cats can play with each other as they roll this. Cats can fish out food. They can chase food. This is ideal for wetter food. If you just want a cheap and easy kibbled solution, you can put little egg containers all over your house with just a few kibbles in them. Anything to give them that tactile and intellectual and nutritional hunting stimulation that they would have had. We haven't mentioned the space needs of cats, but I think we need to. Can we meet the space needs? Well, if you look at the classic McDonald, Knapp's, and Turner and Bateson work on farm cats, they found out that the average cat used 1/10 to 1/2 a hectare. Males used 4/10 to 1,000 hectares, and females used 2/100 to 170 hectares. I think this female never got out of the barn, but that's quite a range. Okay, so more males move further, but these are farm cats. They're fed on farms, they're fed remains, they have a good hunting environment. They're not really kept as pets. They're an intermediate between a pet and a feral cat. When you do the study on owned and unknown cats in one area of the United States, this is where the University of Illinois College of Veterinary Medicine is. If you do this study in that environment, which is not a dense urban environment, but it's certainly not rural, and you compare owned cats versus unowned cats, you find out, regardless of the method you use to estimate home range, you find out that if you're owned, you occupy relatively few hectares. If you're unowned, you occupy quite a few. So in other words, if you need to stop cats from preying on wildlife, owning them will help. So will keeping them in. If you need to protect cats from traffic, owning them will help. They won't range as far. If you need to protect cats from becoming prey, very common where there are coyotes and bobcats, you need to own them to keep them closer so they don't range further. If you're worried about diseases that cats can catch, whether they can spread them to other cats or also spread them to humans, they range further. They're more likely to come in contact with many more diseases. What about vertical space needs? Do we ever think of this? We tend to think of, two-dimensional environments, which is what home ranges measure. But what about vertical space? This is a house in Belgium, very common urban dwelling, and you can see that the windows are open and the cat's sitting on the window ledge watching the person in the garden. So can we meet this cat's three-dimensional environment and its needs for sitting on a window ledge? Well, this is how the cat meets them. This is the cat actually doing this. And all of these wonderful cat pictures that were not taken at the NAVC were taken by Anne-Marie Doche, who's just been fantastic in getting these pictures for me. And if you look at this, this is what this cat does. If you think you can meet those needs with an indoor cat, and you can, but you have to be conscious of it, then you're meeting the cat's needs. But what happens if we fail? Let's see what happens. Hannes Lowy and his group has done a very large survey. Now, this is a survey. They didn't watch the cats. They asked questions of almost 6,000 cat owners of 40 breeds. And they found out that cats that were weaned before 8 weeks of age were at increased risk of aggression to humans. Cats that were weaned later, 14 weeks of age had a lower probability of increased aggression to humans and a lower probability of stereotypic behavior. So here are the data. If you hold on to them for 14 weeks versus early wean them, you really decrease the risk of aggression to strangers. If you hold on to them for 14 to 15 weeks versus 12 weeks, you decrease excessive grooming and other potential obsessive compulsive, ritualistic, stereotypical behaviors. Most people don't hold on to their cats that long. So if we really want to decrease risk, we may have to create environments where the environment travels with the cat or where these needs are met in new homes. So what can we conclude? What are our summary recommendations? For owned cats, simply owning the cat decreases roaming, reduces disease, reduces predation, and reduces accident risk. It decreases stress, and if you can decrease stress and you can have an excellent diet for any owned cat, they will do better. For owned cats, you need early and ongoing intellectual exposure, and I can't emphasize this enough. Most cats could do many more things than people ask them to do and would love to do them. And if you're a breeder or you're advising a breeder, please advise them to select for outgoing dads, bold, not shy moms, to handle the kittens as close to birth as possible, to keep them with the moms and sibs as long as possible, meaning at least 8 weeks, but possibly 12 to 14 weeks. If these are feral cats, step in and do the feeding that they can't do themselves. Feed the mothers excellent diets, enhance them with EPA, DHA, taurine, and a good source of protein. Protect them from stress. Assume that these individuals may be shy or may have been affected by epigenetic effects if they're truly feral or stray. So they may in utero have been affected by epigenetic effects and they may be shy. That's why they end up unowned. Feed the babies from the start. Do everything you can to get a great diet into the babies so that you can pseudo delay weaning. You know, make sure it's an extended period of time you provide this diet and encourage any exploration with novel objects, novel situations, and novel people. And finally, if these are going to remain as feral cats, you have to vaccinate, trap, neuter, and release early, very early, and make sure you use anxiety and pain meds and use that as an opportunity for enhanced exposure and feeding so that you give them the best neurological start possible. Again, you're more than welcome to email me any questions. I edit the journal in the field. This is what it looks like. And for those of you who may be interested in taking the North American Veterinary Institute course, we still have some slots for this year. It's a phenomenal immersion environment. You get to spend 12 hours a day with 3 specialists in the field. We do loads of labs. It's basically a free-for-all Q&A and lectures and hands-on. We do invite veterinary students. They have to have the permission of the course organizer, which is me, but we've had veterinary students. If you don't have exposure and you are a veterinary student, if you don't have exposure at your home veterinary college, and most people don't, to these types of experiences, this may be a benefit to you. This year, we're seeing a series of real cases, so it really will give you an all-around exposure in addition to the lectures and the labs and the questions and the whole environment, and we'd love to see you. So this has been a delight for me. I hope it's been helpful for you, and I look forward to the rest of the series, the rest of the lectures in this series. And again, I'd very much like to thank Alice for inviting me to give these lectures. Thank you, Alice.
Thank you very much, Karen. So let me just wrap up by thanking you formally on behalf of all of us who have been listening to this very fascinating talk. And you mentioned the PDF of the slides, and I do have that available, so I will be sending that out to everyone. And that's it. And thanks very much, and we'll talk to you next week with the dog lecture.
Sounds good. Thank you very, very much.
Okay, I'm signing us off then. Thanks. Bye.
Bye-bye.